Okay, this is a recording and we're going to talk about digestion and where we got up to. So I think we got to the point where we're having a look at peristalsis and what peristalsis is. And I did the demonstration of being upside down and being able to swallow and it being able to push the food all the way up. So we're going to have a look at peristalsis, what it is, and then we'll get into more of the details about the stomach, the small intestine, and the rest of the stuff. Okay, so the first thing is we want to know that in the small intestine there's a cross section, and we're going to have to label this as well. But if we look at the cross section of the small intestine, there are layers in it, and a bit later on we'll start drawing the villi, which are in here, the finger-like projections. But now we're just thinking about what's happening in these two layers. So if I draw all the way around, so one of the layers there, this layer, the sort of outer layer, this is the longitudinal. Longitudinal muscles, and what they do is they're going to contract, they're going to shorten in this direction, they're going to pull in. And so what this does is if they both contract, then overall the whole tube actually becomes smaller, at least if we consider this section here. When these contract, it's going to move. And that means that if there's some food which is going through, now it doesn't have to go through so much. It doesn't have to travel through the whole distance. So the longitudinal muscles contract, and that shortens the length. But that wouldn't push the food through. So we need some kind of force from behind. And this is where the next layer in, this layer here, the circular muscles, what these are going to do, muscles, what these are going to do is contract. So if we think about it being a circle, the contraction is happening towards, it's all the way along. So all the way along, this is going to shorten by contracting in all these different sections. This section's contracting towards this section, so all the way along. And what that's going to do is make a smaller circle. So if we think about that there, if we made a smaller circle behind, then it's going to be kind of squeezing in. It's going to push the food through. So our circular muscles and longitudinal muscles work in a wave. So the circular would contract here, and then the longitudinal would be contracting ahead to then push it through. Then they would relax, and then the food has moved through. So let's just go back to that. So yeah, it's helping the food to move along, and it's helping it to move through. And then also if you've got fiber, that's also going to help. Okay, the next thing we want to think about then is what's happening in the stomach. So in the stomach, we can draw, going back to our picture of the stomach, we think of it as being this kind of J shape. It's kind of a J-shaped pouch, which was then going to lead to, just a reminder of these junctions here, it's going to move to this T junction. So we've got that T junction, our next T junction, and then going up behind, and then it's going to do the Y, and this is where our liver was. And then we've got the pancreas here. But we're going to think about particularly what's happening in the stomach. So as the food comes down, it will be detected. So in this wall here, there are lots of receptors. They are sticking out. So if this is our cell, they are sticking out at the top, and we can have food molecules that are going to go and bind to that receptor, and that's going to stimulate the stomach. So it's also when we see, this is my eye, if we see food, also if we smell food, then we're also going to get nerves coming down, and we'll deal with this in the next bit. Nerves come down and they're going to tell the stomach to start to secrete things. So within the wall of the stomach, there are a number of things, and one of them is a pump. So we've got pumps, which are going to pump in H+, proton pumps. So they're going to pump that in, so we're going to get hydrogen ions, protons floating around. We've also got chloride pumps, so they pump in Cl. And then we're getting our HCl, our hydrochloric acid, which drops the pH to about pH 1 to 2. So once it's an acidic environment, we've got other proteins which are going to be released. And so out of cells here, we've got the release, the secretion of an inactive form. So what gets released is pepsinogen. So in bio, whenever we see this gen, it means it can generate. So pepsinogen, it means it can generate pepsin. So what happens with the HCl, pepsinogen folds into the right shape, takes the right conformational shape, and it becomes pepsin. And now this, the pepsin, is the active form. So now we've got an active protease. And that active protease will start to digest. So if we remember, we had the idea of protein structure, a 3D, the tertiary structure of a protein. And what the protease will do is it will come and make little cuts. It will chop at various points. And so what we'll get is chains, and they wouldn't be straight, but you get the idea. We've gone from a long protein, chains, shorter polypeptides. So we've gone from protein. Now by making cuts, this protease, it's gone into shorter polypeptides. Okay, let's just go back. So yeah, we talked about the low pH environment. We've talked about the idea that we've got things which are being secreted, and those are by glands which are called gastric glands. The low pH is going to start to denature the proteins. So we talked about, if we go back, we talked about that pH is one of the factors that affects enzyme activity because they're proteins. So if we've got a low pH, what's going to happen is all these H pluses are going to go in and they're going to interrupt the bonding here. So we start to lose shape. So it will denature proteins, but H plus doesn't digest fully. So for that, we need our protease enzymes. Yeah, and it's a muscular pouch, so it's churning around. The mechanical action is going to mix the juices with it, with our active protease. Our low pH is also going to kill bacteria and other pathogens. So pathogens are killed by it, until eventually it's going to start to leave into the small intestine.